All right, Alex, you about ready?
Yeah.
Okay, well, greetings, everyone. Welcome to our monthly Pathways Research Symposium. We have two terrific speakers today. Our first presentation is going to be given by Alex Kostic from Joslin. His is an initiator award that he's had since 2017. And it's about gut microbial fermentation, products of muscle-derived lactate as mediators of exercise and metabolism. As always, please use the chat to ask questions, and I'll moderate after the presentation. Alex, take it away, please.
Great. Thank you so much, Chris. And I'm really excited to tell you about my work today. Oh, is this showing a blue screen right now?
It is, yes. That worked a second ago.
How did that happen? Yeah. Sorry.
We did test.
Yeah, we did the test too. Let's try this again. How's this?
Oh, there, now it's there.
All right, great.
Thank you.
Yeah, really excited to tell you about my work, and it's been such a privilege being part of the Pathway program for these past almost 5 years. You know, encompassing the time in starting my own independent lab, and starting with the Pathway Award has made such a big difference in being able to tackle really big questions. In my mind about what's at the heart of microbiome interactions with human physiology, in particular its relation to diabetes. So first, just a disclosure, I'm a co-founder and on the SAB board for FitBiomics, which actually is a company that spun out of the discoveries developed here. So my lab takes kind of a broad approach to understanding the communications, the interactions between the human microbiome, and particularly in the gut, and its interface with immune cells, and also just generally with metabolism. And of course, we're very interested in and focus our studies around models of type 1 diabetes and type 2 diabetes, respectively. So the award has allowed me to develop a program that's expanded in many different directions, and perhaps too many directions. But I want to tell you about one story that's come out of this that is particularly exciting for me, because it looks like it's going to be my first funded R01, which is great timing as I'm kind of moving out of the Pathway Award, and so it involves understanding the interaction between microbiome and metabolism. And just a little point to make here is I always like to think of all the work that we're doing in the microbiome as what is the relationship between this microbe, or the whole microbiome, and the human. Right now there are no really highlighted examples of mutualism in the human gut. That's the example being a bee and flowers, that kind of a relationship, and so, but I think that there's a lot of evidence and accumulating, and I think one thing maybe to keep in mind as I go through this is I think what we're describing here kind of fits into what I think is a real mutualism between microbes and human metabolism. So this. I'm always a little bit. I wish I started with a better hypothesis. I come from the Broad Institute. Both my PhD and postdoctoral training were at the Broad Institute, where we don't have hypotheses. Now these days I'm into hypotheses, but literally where we started was trying to understand what are the unique aspects of the physiology of very healthy individuals, metabolically very healthy individuals, and in this case we're studying runners in the Boston marathon and collecting various aspects of their health, but of course with a focus towards the microbiome. And so we collected from volunteers microbiome samples before the marathon and after the marathon, and compared these to microbiomes from healthy volunteers that were collected around the same time. And so one interesting thing that we saw was this bloom of a microbe that I'd not really heard of before, Vionella, genus Vionella, that goes up in abundance in marathon runners relative to sedentary controls, and stool samples. You can only produce one per day, maybe even less often. So you start seeing the bloom after the marathon day. So that was kind of peculiar. When we looked at the abundance of Vionella in these individuals, and on the left are the marathon runners, right are the sedentary controls, and we're looking at samples before the marathon and after the marathon. One thing that jumps right out is just how much more abundant the Vionella is than the marathon runners in general. It is still present in the sedentary controls. You can see there are some very small abundances there. And also, again, this blooming of Vionella, but it's also, again, more abundant pre-marathon in the marathon runner. So, what's going on here? Maybe some interesting biology. Looking into this microbe that I never heard of, it's a obligate anaerobe gram-negative cocci, nothing special, but it is kind of relatively unique or unusual in the gut, is that its sole carbon source is lactate. As in the lactate that's produced in abundance during endurance exercise. And I'll go into the biochemistry a little bit, but it metabolizes this lactate into short chain fatty acids, principally propionate and acetate. And so on a validation cohort of ultra marathon runners and some Olympic trial rowers, we did deep shotgun metagenomics, and this time focusing on, we saw Vionella again enriched, but this time focusing on the individual microbial genes and their abundances, before and after strenuous exercise. And apologies, it's a little bit hard to see, but what I'm showing here is the full pathway of lactate metabolism into acetate and propionate as it's represented by Vionella and also other lactate utilizing bacteria. And the really striking thing that we saw was that every single enzyme in this pathway was higher abundance in red after exercise relative to its abundance before exercise in blue. So this shows that the microbiome is responding at an enzymatic level to exercise, and could it be responding to lactate? And so we asked that question. So it wasn't known before whether systemic lactate actually makes its way into the lumen of the gut. And so we did IV injections of labeled lactate in mice, looked at the serum plasma and cecal contents and colon contents. And what we found was that, obviously there's a lot of it in the serum and plasma 12 minutes after injection, but it's also present in the cecal contents and the colon contents. So we're slicing it open, sampling very carefully just the contents, not actually trying to sample the tissue. So it gets there apparently, and so then the it's a question of sufficiency and trying to understand causation, rather than correlation. Can this microbe improve endurance exercise? And so, again, the way we went around this, we isolated one strain of this Vionella atypica in particular from an athlete. We start introducing it into mice and run them on a treadmill. And this work was done in collaboration with a really excellent exercise physiology lab, Sarah Lessard's lab, right next door to mine here at Jocelyn. And so we gave the mice by oral gavage either Veillonella atypica or a control. It's always hard to choose a control. People often do just PBS, but then that doesn't control for bacteria. This control that we use, Lactobacillus bulgaricus, is a simple yogurt bug that critically cannot use lactate. Actually, it's a lactate producer rather than consumer. And so we did a crossover design in this study. So one group was given one treatment, the other the control 1 week, and then 5 days later we do a washout and then switch and then look at the results. And what we found was, of course, this is a noisy assay, but a significant increase in runtime in the mice that received the Veillonella, and that's both the groups before and after the washout, the ones that had the Veillonella perform better. And so how could this be happening? What's the potential mechanism? And of course, this is where microbiome research always gets really challenging. But so I've mentioned that Veillonella can metabolize lactate into propionate and acetate, which we think is important. One hypothesis was, can it work as a lactate sink? And could that be a mechanism where it's active? But that's not likely. Just currently the understanding in exercise physiology is that buildup of lactate in muscles, contrary to public belief, is not what causes fatigue or pain, not the lactate itself anyway. And so what we did was a simple study where we just intrarectally instill propionate right where it's being produced by anaerobic gut bacteria in the colon. 30 minutes prior to running and run these mice, and we get to see a effect size that recapitulates a whole colonization with Veillonella, this increase in runtime in the propionate treated mice. Okay, so that's kind of interesting, but what does propionate do when it gets there? What does this have to do with exercise? And so to start tackling that question, I pondered, do muscles actually take up propionate? And for this, I reanalyzed some really amazing data from Joshua Rubinowitz's lab, where he looked at metabolite exchange across many different organs in pigs. And the idea here was sampling venous blood and arterial blood across many different organs, and then measuring the ratio of any given metabolite through metabolomics in the venous versus arterial blood, and that's how you can get a sense of whether a metabolite is being uptaken or produced by a given organ. And of course, we're looking at these short chain fatty acids. But by the way, propionate and butyrate, when you see them in the serum, they can only be produced by the gut microbiome. Acetate can be produced by endogenous metabolism as well. But human propionate and butyrate, that's coming from the microbiome. And so looking at this, we can just kind of plot out. These are all the metabolites that they looked at. We can plot out the relative degree to which the colon is releasing them or taking them up, and then the same with muscles. And so just to guide you, as you'd expect, glucose is taken up by both colon and muscles. Lactate is of course produced by muscles, and this is at rest, but apparently also produced by the colon. So all three short chain fatty acids, butyrate, acetate, propionate, are produced by the colon, coming from the microbiome, and they're all taken up by the muscles, actually. So these short chain fatty acids seem to be going into muscle tissue. So what do they do when they get there? And thinking of propionate in particular, So short chain fatty acids have a GPCR that they can signal through. Actually, two of them, FFAR2, FFAR3, and there are actually a couple of more, but that's the main ones for propionate and butyrate. And so we asked the question, do we see evidence of this happening, signaling in muscles with treatment of propionate? So first, this is just showing that these receptors are robustly expressed in mouse skeletal tissue, and then when we treat C2, C12 myoblasts with propionate. Actually, no, sorry, this is not treatment. This is just differentiation of the myoblasts. And actually, as they differentiate into myotubes, you can see the expression of these receptors starts increasing. And then when we treat the differentiated C2, C12s with propionate, we see this increase in activated calcium calmodulin dependent kinase, which signals to AMPK. And so we see this activation of AMPK in muscles with propionate. And so this is important because AMPK has a lot of functions in muscle tissue. It increases glucose uptake and storage, fatty acid uptake and oxidation, fiber switching to an oxidative type and increasing mitochondrial content. The other way this might be acting is by direct import of short chain fatty acids into the cells and then oxidation in the mitochondria. And so we did a seahorse experiments. We starved the cells of myotubes of glucose. We add in either PBS or propionate and show that yes, propionate can be oxidized by muscle tissue. And so what does this all mean? So for why do we depend on microbiota for exercise? And we do to some degree. We know that studies before ours showed that germ-free mice and also mice treated with antibiotics have decreased running endurance compared to regular colonized mice. So we are hunter-gatherers, or we have been for 50,000 generations, except for the past dozen or so. And so one important part of hunter-gatherer lifestyle, in addition to foraging a lot of high fiber foods, which I won't go into, but is hunting, and endurance hunting in particular. We can't catch this antelope. It's too fast, but we play the long game. We start a day at 8 a.m. and we run and we run and we run until 3 p.m. when we've reached 35 kilometers. And the gazelle is so tired, and so apparently hunter-gatherers like to target males because of their big horns. They just collapse, and that's when we take them. And so we have a method of recycling lactate produced by muscles. It goes back in the liver and undergoes gluconeogenesis. But how much more efficient would it be if this product of glycolysis it goes into the gut, where it's metabolized for free to us by our gut microbes that convert it into short chain fatty acids such as propionate, and then these short chain fatty acids go on to have these effects on muscles, including direct oxidation, serving as a fuel source. So as lactate accumulates, microbes begin doing this, and so. Just to wrap up, I promise all of this is related to diabetes, and that's the focus of our future work. So this is a study, a really fascinating study. We're involved at all in this, but I reanalyzed some of the data And so here they're looking at overweight men with prediabetes. They put them into a 12-week high-intensity exercise training regimen. And then afterwards, they notice that most are responders, but some are non-responders in terms of improvement in insulin sensitivity. And I'll tell you about that. So most people lost weight and all lost fat mass, of course, it's pretty intensive exercise regimen. But what's really interesting is that only responders had improvements to glycemic control and insulin sensitivity, which is really terrible, I think, in diabetes. Here is a tool that you're supposed to use to help improve your metabolism, but in some people it actually makes things worse. And so this is known as exercise resistance. And so what we found in this cohort, they had gut microbiome data, and what I could see in Bionella alone, just one example of a lactate utilizing bacteria, is that in the non-responders, the abundance of Bionella actually went down after this 12-week training period, whereas in the responders, it stayed the same or even went up. Then looking at short-chain fatty acids in the blood of these individuals before and after the 12-week regimen, nothing important is happening in acetate. If we look at propionate, we see this big increase in propionate in the blood after this 12-week exercise regimen, only in the responders. Butyrate, we see this again, this big increase in butyrate in the serum after exercise, only in the responders. And in fact, in the non-responders, butyrate in the serum goes down. And so we're very interested in this problem and how introduction of lactate utilizing bacteria or short-chain fatty acids themselves could improve the situation. And so in summary, I've shown you that lactate coming from muscles during exercise is being metabolized in the gut microbiome by lactate utilizing bacteria into short-chain fatty acids that then feed back into the muscles and have some positive effects, including in glycogen storage, oxidative capacity. And some of the next steps we're looking at, as I already mentioned, is seeing whether lactate utilizing bacteria could be helpful in this in exercise resistance. Trying to really understand some of the pharmacokinetics, basic pharmacokinetics of is muscle lactate, really a dominant? Can it be a dominant source of short-chain fatty acid production in the serum? And trying to better understand the mechanisms by which short-chain fatty acids influence muscle and metabolism and function. And so with that, I'd like to thank the many people in this lab involved in this work, mostly students, some of which are now graduated, and my collaborators. Thanks very much for your interest.
That was really cool. Thank you very much. Really nice to see it come together at the end with that human study. You must be pretty excited about that.
For
so
sure.
I had a question about the mouse studies. When you are looking at your propionate producing lactate consuming bacterial strain, are you putting that in and getting an effect on exercise performance into germ-free mice? So in other words, it's the only bug you're putting into the gut, or are you putting it into a pre-existing normal bacterial culture when you do that experiment?
Into mice, just SPF mice, or in our barrier facility. We have not done notobiotic experiments yet.
Okay.
We wanted to do it that way, just to test how relevant it is. Sometimes, notobiotic experiments can be a little bit too artificial. And so we thought it was important to look for effect in the context of a wider kind of complex microbiome.
Okay, thanks. Let's see. What I am, oh wait, hang on one second. Why am I not seeing? Oh, here we go. All right. So first from Jane Royce, you mentioned that you were looking at diabetes models. Do you see a correlation between circulating propionate and receptors?
Yes. As to whether it changes the abundance of or expression of the receptors, that's a good question. We haven't looked at that. But the short chain fatty acid receptors, FAR2 and 3R, are expressed on mice on muscles, and we are seeing signaling with propionate.
Yeah. Okay. So, yeah. So the question was whether conditions that raise the propionate also enhance the expression of those receptors, and you haven't looked yet.
We haven't looked at. Yeah.
Okay. All right. So Ernesto Bernal Mizrachi, cool data. What's the effect of metformin or other diabetes medications on propionate production by the microbiome?
Great question. Metformin, some of its effects seem to be very highly interactive with the microbiome. I don't know, but in this study that we showed, the human study, that was all drug naive individuals with prediabetes. So at least it does something in the absence of metformin.
Okay, Marlon Pragnell, very nice work. I noticed Valium levels, I don't know if I pronounced that right, sorry, your bug, went down in non-responders. Do you have any idea why? That's a good question.
No, I really don't. That's one of the things that we'd like to understand. I mean, presumably these people are exercising, producing a lot more lactate than they had before once they're, and so why isn't their Vinella blooming in response to this? I don't have a good answer to that, but that's a great question.
Okay, and Alan Addy, how does lactate get from the circulation to the gut lumen? And how much of the propionate goes to the liver for gluconeogenesis versus goes to the muscle for what you're describing?
Excellent questions. Excellent questions. And I think the liver is going to be particularly important because of the portal vein feeding right into it. One of the nice things about the lower gut, which is where Vinella and these anaerobes tend to hang out, is that especially more in the rectum, the metabolites there are more systemically privileged and can bypass portal metabolism. But that's something we haven't looked at, and I think is important, because if the vast majority is being metabolized in the liver, that doesn't leave a lot to the muscles. But really great point.
So Chuck Grant asks, why do you say propionate is only produced by bacteria? Oxidation of isoleucine and valine produces propionate. I think actually.
Yes. So it is true. Yeah, in beta oxidation, you do get the formation of this propional acyl CoA.
Propional CoA. That's right.
Produced endogenously. But the difference, I think, with the microbes, is that this is their primary end product. And so if you look at the serum of mice, of germ-free mice, they do not have any detectable, propionate, butyrate. Similarly, if you knock down with antibiotics mice, you see a reduction. It's their primary end product, whereas for us, it's kind of an intermediate. And probably someone knows that better than I do. And please do correct me if I'm getting something wrong.
And I think we'll take a last question from Chris Rhodes. He speaks of, again, downstream propionate metabolites, propional CoA, propional carnitine. These change in NAFLD-NASH. So I think the question is there a potential connection there?
That's really, I didn't even know about that. So I don't know, but that's a great point. I'll look into that.
And actually, let me squeeze one more in for my homeboy Phil White. Do you know whether certain dietary constituents could be employed to amplify the effects of exercise on this mechanism via effects on microbiota composition? That's a big question.
I would imagine using fermented foods that have lactate. A lot of microbes produce lactate. So now potentially you're consuming a lot of lactate that's going to help bolster your collection of an abundance of your lactate utilizing bacteria in the gut. Just one idea, but not tested.
Okay. Well, just you can tell from the questions, everybody was pretty excited by that. Thank you, Alex. Terrific talk. We'll move on to our second presentation today, Sui Wang from Stanford. Also an Initiator Award recipient beginning in 2016. And now a completely different topic, protecting the retina from diabetes-induced damage. Sui, please.
Hi, thank you for the introduction. First, I would like to thank the Pathway Program for the generous support. Sorry. Go back to my. Okay, I'll just continue. And this award is instrumental in helping me transition from a postdoc fellow to an independent PI. So I was trained as a developmental biologist, so focus on study transcription factors and transcriptional regulation. So I had a research experience study pancreatic development and also retina development. So as I was transitioned from postdoc to PI, I decided to merge my expertise on diabetes and retina biology to study how the neural retina responds to diabetes and how we can protect the retina from diabetes-induced damage. So today I will briefly share two unpublished stories. Any feedback is greatly appreciated. So as shown here, diabetes can induce retinopathy. So diabetic retinopathy is one of the most common complications of diabetes and the leading cause of blindness in working age population. So this disease progresses from early non-proliferative stage to late proliferative stage, and eventually can lead to vision loss. And then current treatment strategy focus on or target the late proliferative stage, and including the treatment, including anti-VEGF drugs and steroids and other surgical techniques. However, those treatments can delay the development of the disease, but it cannot restore vision loss. So we ask whether we could protect the vision under diabetic conditions by blocking the initiation and the early progression of this disease before vision is compromised. And then to achieve this goal, we first need to understand how the retina responds to diabetes in the early stage of diabetic retinopathy. So here shows a schematic of the vertebrate retina, in case you do not know the retina. So it is composed of multiple different types of neurons and also glia cells and vasculature. So there are more than 100 different cell types in the retina, and different types of cells respond to diabetes in a different fashion. So for example, the photoreceptor cells, which are the cells which are the light sensing neurons in the retina, they undergo, they become like this, they have dysfunction. And then the glia cells, amyloid glia, which is a major glia cell type in the retina, undergo reactive gliosis. And ganglion cells, which is the projection neurons in the retina, they undergo degeneration. And we also have vascular lesions and microglia activation in the retina in response to diabetes in the early stage of diabetic retinopathy. But the molecular mechanisms that underlying these responses are not like clear or fully elucidated. We aim to uncover the molecular underpinnings that are in charge of or regulate these responses, with the hope to develop like novel therapeutic strategies. So we use streptozotocin-induced diabetic rats as animal models. This STZ rats has been widely used in the field to study early diabetic retinopathy. So we first confirmed the retina phenotypes induced by diabetes in our rat colonies. So we first show that we found that at 2 months after STZ injection, the major glial cell type, Mueller glia in the retina, undergo gliosis, indicating here by the expression of upregulation of GFAP. You can see it's very clear. And at 3 months after we induce STZ, we inject STZ, we see a significant reduction of the number of retinal ganglion cells. So here's a quantification, and especially in the peripheral retina, which is consistent with the situation in human patient. And at 5 months after STZ injection, we see some vascular lesions in the retina. So here is a staining of desmin, which labels the smooth muscles, vascular smooth muscles and parasites, and its expression is significantly downregulated on retinal vessels, as shown here in red. This result suggests that the STZ rats, they develop neurovascular abnormalities in early stage prior to the occurrence of severe vascular lesions, which is consistent with the existing literature. And to uncover that diabetes-induced multicellular responses within the retina, we decided to apply the single-cell RNA-seq technology to diabetic rat retinas. So for those of you who are not familiar with this technique, so the single-cell RNA-seq can profile the transcriptomes of thousands and millions of single cells within the tissue at once. And based on the gene expression profile, they can be clustered into different types. So we can systematically look at the profiles of multicellular responses induced by diabetes in the retina at the transcription level. So what we did is we induced diabetes in rats, and we harvest the retina at 1 to 5 months after STZ injection. And after we harvest the tissue, we rapidly dissociate them into single-cell preps and submit them for single-cell RNA sequencing. So here shows the result from control and diabetic retinas at 1 month after STZ injection, which is at a very early stage. At this stage, no obvious retinal phenotypes can be detected in our animals. But however, you can see that there are changes already happening at the transcription levels for some of the cell clusters. The most rapid change has been seen in retinal Mueller glia cells. The wild type and STZ retinal Mueller glia cells start to separate from each other, suggesting their transcriptome become very different in response to diabetes. And we check the genes that are differentially expressed in Mueller glia cells at the very early stage between wild type and STZ. We found that majority of the genes that are upregulated are actually protective genes. So showing here is the heat plot showing the gene expression level in STZ over wild type is the fold change. And then the hot color means it's the fold change is much higher, and then the cold color means the fold change is lower. And each of the rows or cluster of genes can be recognized as anti-apoptosis or anti-oxidative stress genes. And the columns are different stage after STZ injection, different months after STZ injection. You can see that for the anti-apoptosis, anti-oxidative stress genes, they were rapidly upregulated in Mueller glia cells 1 month after we induce diabetes, and their expression upregulation goes down and goes down and even below the normal expression level. And then the same situation for anti-inflammatory genes. And also we see genes that are known to promote adhesion of endothelial cells. They start to be upregulated at around 3 months after STZ, and then goes down at around 5 months after STZ injection. So based on this, we hypothesize that Mueller glia cells upregulate protective genes, but they fail to maintain this beneficial effect in response to diabetes. Then we wonder if we artificially or by different methods, if we can sort of maintain this upregulation of this beneficial protective pathways or protect the genes, can we protect the retina from diabetes-induced damage? To test this hypothesis, to test this hypothesis, we first start with a single candidate, which is ZAP30, which is ZAP3016. It is within this cluster. It's an anti-inflammatory gene. And we first confirmed the expression pattern of ZAP36 in the retina at different stages after we induce diabetes, after STZ injection. By either quantitative in situ or qPCR. Unfortunately, the antibody doesn't work so well for us. So here's the quantitative in situ. So at different stage, we dissociate the retina into single cell preps. And then the red color is the mular glial cells labeled by SOX9 antibody staining. And then the green is a quantitative in situ. So each dot is equals to mRNA molecule. So by counting the number of dots within the cells, we can have an idea of the expression level. So the top panels are wild type at different stages, and the bottom panels are STZ conditions. You can see the expression level of ZAP is significantly upregulated starting from 1 month after STZ injection, and it goes up more at 2 months, and then goes down after 3 months after STZ injection. So this confirmed our finding in the single cell data, and also we confirmed that by qPCR. And I don't have time to show you, but we also have this confirmed the expression pattern of ZAP36 in human diabetic retinopathy tissues. So then we ask, what's the function of ZAP36? We generated a AAV, which drives the expression of a shRNA construct that can specifically knock down ZAP36 expression. And we use a MG1 promoter, which we identified in the lab. It only drives expression in mular glial cells. We are not going to target or disturb any other neurons in the retina. So we inject the virus and then induce inject STZ in the rats. And then after 2 months, we harvest the retina. So we first check the gliosis of mular glial cells. So here are the top panels are the control, and then when we have STZ injected, you can see the retina morphology is significantly disturbed, and you see a huge increase of the gliosis indicated by the expression of upregulation of the GFAP shown here. And we also detect a significantly increased degeneration of retinal ganglion cells. And also we see at 2 months, usually we do not see vascular leakage at 2 months in the retina after STZ injection. But right now, if we knock down ZAP36 gene, we do see the leakage or lesion of the retinal vessel. So NHS biotin is a small tracer molecule. It's about like 200 kD. So we see those molecules get leaked into the outside of the vessel in the retina. So this confirmed that this expression is required probably for protecting the retina from diabetes-induced damage. And we further ask, if we overexpress this gene, can we achieve a rescue effect or protect the retina? So we generated another virus, overexpressed the ZAP3016, and we also inject this AAV into the animals and harvest the retinas at 3 or 5 months after STZ injection. And here we see that the gliosis is significantly suppressed by upregulating ZAP36. And also at 5 months after STZ, we do see the rescue of the desmin staining, which desmin staining the vascular smooth muscle and parasites, which is indicating of the integrity of the vasculature. So it seems we can rescue the phenotype. So in conclusion, for this part of the talk, we think the ZAP36 is upregulated rapidly in mular glial cells by diabetes in the retina. But however, the glial cell cannot maintain this upregulation. So the level goes down after 3 months after STZ injection. And we think failure of this maintaining the ZAP36 levels contribute to the development or initiation of diabetic retinopathy. I don't have time to show more data, but we believe that the ZAP36 is a strong inhibitor of inflammatory responses within the retina, and that probably are the downstream mechanisms. And we also have identified a few targets of the ZAP36. So just to give you a big picture of what we are we got from this ZAP36 focused study. Okay, so it's obvious there's not only one gene is upregulated, or not only one protective genes are upregulated by diabetes in the retina. There are multiple genes has a similar pattern, or multiple protective genes has similar expression pattern. So they're upregulated by diabetes, and their levels cannot be maintained as the disease progresses. So we were wondering, can we upregulate or activate multiple protective genes at once, and whether we by doing this, whether we can achieve even like a further protection of the retina from diabetes. And to do this, it's not easy because co-transducing two different types of AAV into the same cell in vivo, the efficiency is low. And the co-infection rate is even lower if we want to co-transduce 3 or more than 3 AAVs into the retina cells in vivo. So to solve this problem, to achieve a multiplex gene activation in vivo, so we turn to the CRISPR system. And so it's well known that the endonuclease dead Cas9, if we Cas9, for example, if we fuse the protein with VPR, which is a transcription activator, and it can specifically activate genes, endogenous genes, activate the transcription of endogenous genes. And then however, the Cas9, the commonly used Cas9 protein, it can only process one guide RNA, which means you can only activate one gene at a time. So that not serve our role. Like we want to activate multiple genes at once. So luckily the DCAS12A, which is a nucleus in the CRISPR in the Cas9 protein family, a Cas protein family called DCAS12A. This guy is able to process a guide RNA array with multiple guide RNAs target different genes at once. However, the problem is the wild-type DCAS12A has fairly low efficiency in vivo, so it's not useful for us. So to solve this problem, we established collaboration with Dr. Stanley Chi at Stanford Bioengineering, and we designed systematic screenings to identify DCAS12A mutants, which allow for efficient or high efficiency gene activation and also multiplex gene activation in vivo. So specifically, what we did is we examining the structure, the residue, the negatively charged residues that are close to the DNA target. So here shows the negatively charged residues and also the DNA close to the DNA. So we systematically doing the mutations. So we hypothesized that if we change the negatively charged residues to positively charged arginine, and that may increase the binding affinity of Cas12A to the negatively charged DNA. So we mutated a lot of the residues and then use a cell culture system to screen for the mutant has the best efficiency. So the system is showing here. So we generated a cell line which expressed GIP under the control of TAT0 sequence repeats. And then we also generated a construct, express the guide RNA, target the TAT0 sequence, and then we co-transfect two plasmid. One contains the DCAS12A mutant, and the other one contains the guide RNA. And then we transfect into the cells, and then we examine the expression of GFP to see which mutant gave rise to better activation of GFP. So here shows the result. It's a busy slide, but I will go through it like one by one. So here I want to mention that we also have a GFP expression, BFP expression construct after the guide RNA. So this can serve as a transfection efficiency control. So for the BFP positive cells, you can see some of the mutations, residue mutants, they can significantly upregulate the GFP expression, activate the GFP expression compared with control. And interestingly, if we only select the cells express low level of BFP, which means the expression level of guide RNA are probably also lower. So it has low concentration of guide RNA. And then we see that in the for wild type DCAS12A, it cannot sort of robustly activate the GFP expression, but some of the mutant can still, even with low levels of guide RNA expression, can still significantly increase the gene, activate the gene expression. And we did combinations of different mutations and eventually identified this or made this Cas12a, which we named as very good D-Cas12a, VGD-Cas12a, and has it outperforms all the D-Cas12a mutants and has great efficiency in activate GFP expression. We first examined the expression of the system, see whether it can achieve multiplex gene activation in cell culture system. So we designed a guide RNA array targeting the Yamanaka factor SOX2, KLF4, and ARC4. We transfected it into the cell culture system. Compared with non-guide RNA control as well as wild-type D-Cas12a, the VGD Cas12a can significantly increase the expression or activate the expression of the Yamanaka factor simultaneously. And here is a quantification. So then we tested the multiplex, the gene activation efficiency of VGD Cas12a in the retina in vivo. So we made a construct containing the OSK guide RNA array together with the VGD Cas12a, and also co-electroporate another plasmid CAC-CFP into a P0 mouse retina pups in vivo. And we let the animal grow for 21 days, and we harvest and examine the retina at P21. So we have so here shows the result. So compare the bottom panels are the controls. So and then each of the big column is standing for the OC4, KLF4, or SOX9. So the retina in the wild type situation, the retina does not express KLF4 or OC4. It's negative in the retina. Retina express a low level of SOX2, but when we have the guide RNA array, you can see SOX2, KLF4, and OC4 are all upregulated by this system. So the results suggest that the system really works and has pretty good efficiency in vivos for us to achieve multiplex the gene activation. So we are so just a summary of this technology. So we identified this VGD Cas12a system, which can achieve multiplex the gene activation in vivo in at least in the retina has a very high efficiency. I want to mention that in addition to gene activation, it can also achieve gene editing or gene repression. And also the advantage of this system is that its efficiency is pretty high when the guide RNA concentration is low. So I think that's an advantage for in vivo study. So currently we're trying to integrate the system into AAVs and use it to test whether we can further protect the retina from diabetes-induced damage by multiplexing or multiplex activation of multiple protective pathways. With this, I would like to thank the people who did the work. So Dr. Cheng Hui Lin is a postdoc in my lab. He's the driving force of the project. And Dr. Stanley Chi, Lucy Guo, and Jing Bin are our collaborators at Stanford Bioengineering. They helped us develop the CRISPR, the novel D-Cas12a system. And Dr. Patrice Ford, Thomas Gunner, they are diabetic retinopathy experts in University of Michigan and provided tremendous support. I also want to thank the PASSE program for funding, and we're also funded by the NIH. Thank you.
Wow, another really exciting talk. Thank you very much for that.
Thank you.
I'm looking for questions in the chat. Alan Addy, we got several coming here. Alan, sorry, it slipped by me. Here we go. Is RAGE a target of ZFP36? Does RAGE expression correlate with inflammation in these cells?
So in our single cell sequencing result, we did not see RAGE. So there's a caveat of the single cell RNA-seq analysis because it's a single cell level of RNA-seq. So the depth is shallow, so we cannot capture all the targets. So for ZFP36, when we did the focus study, we did a bulk, so we sort out the cells and did the bulk sequencing. We also did not see VAGE as a major or top hits. I cannot rule out the possibility, but we didn't see its top hits in our study in the retina, especially in the myeloid glial cells.
Okay, thanks. David Antonetti asks, or says, first of all, exciting story. Does targeting the ZFP alter expression of vascular factors known to be involved in the disease processes, such as VEGF, Ang1, 2, or inflammatory factors?
Thank you. That's a great question. So we did not see the changes for VEGF, Ng1 and 2 at the stage we are examining, because we are focused on pretty early stage. But we do see other factors, secreted factors by myeloid glial cells that are changing, which can regulate vessels. So myeloid glial cell interacts with the vessel, so its processes wrap on the vessel. So they do secrete factors that are interact or changing or affect the vessels. And at the very early stage, it's those factors instead of the VEGF or Ng1 or 2 that are changed by ZFP. But eventually, I think as the disease progresses, I think it affects probably will sort of slow down the disease and also affect the VEGF or Ng1 or Ng2 activation at late stage, I guess. Yeah.
Okay, Michael Dennis, does gliosis precede the early adaptive changes in MG gene expression, or is it associated with perhaps a consequence of the secondary switch in gene expression?
I think, to be honest, I do not know the answer. I felt like it probably associated with gene changes. So it's probably happening at the same time in a pod. And then, so we are, right now, we are just trying to identify the genes which may be beneficial. So glial cells can activate beneficial genes or genes which are detrimental. So we're trying to identify what are the genes that play beneficial roles and what are the genes play detrimental roles, and try to tease out the bad and good effects and enhance the good effect and suppress the bad effects. Do I answer your question? Okay.
Barbara Khan, is cellular senescence involved in the progression of retinopathy? Is senescence underlying the anti-apoptotic effects in your RNA-seq? And does the release of senescence-associated secretory factors contribute to the inflammation?
Yeah, that's a great question. I think in the literature, the senescence is involved in the progression of diabetic retinopathy, but unfortunately, I'm not an expert on that. So, but there are literatures showing that it's involved in the disease, but we did not detect any of the factors related to senescence or non-senescence genes in our single cell sequence RNA-seq or bulk RNA-seq. My suspicion is maybe, I may be wrong, but my suspicion is maybe some of those factors are regulated at the post-transcriptional level, because we're only doing the gene, looking for or searching for the genes that has transcriptional changes in response to diabetes. And then what's the last? So
yeah, so I'll ask the last question if you don't mind. We're at the top of the hour.
Sure.
So really impressed by your ability. If you block the reduction in ZFP36 in your STZ model, then you get nice retention of of structure and function of the retina. What do we know about the function of ZFP36 protein that may relate to that phenotype? And connecting the second half to the first half of your talk, are any of your genetic engineering methods potentially a therapeutic avenue, for example, to target ZFP36, given that you have nice evidence that it could be therapeutic?
Yes, exactly. Yeah, we're trying. So for the first question, we think ZFP36 is a strong inhibitor of inflammation in the retina. So it is known to be a strong inhibitor of inflammation in macrophages. And we show that it's also expressing neural glia cells. Previously, it's not known this factor is expressing neural glia cells in the retina. We show that it's in the glia cells, and we think it's inhibit multiple pro-inflammatory genes at the same time. So it could be like has a more strong effect than if you just suppress single pro-inflammatory genes. And for your second question, yes, we're trying to see whether we can develop this into therapeutic strategies. So the CRISPR system, the VGD Cas12a system, can be placed in AAV. So, and then the retina is an immune privileged organ. So it allow us to inject or do gene therapy or inject AAVs into the retina to see whether we have rescue effect. But my only concern is because we're showing the effect in very early stage of diabetic retinopathy. I'm not sure whether we can find patient who willing to, or find like subjects who can sort of take this thing as early as possible. So I'm not sure whether if we do the therapy at a little late stage, whether that still helps. So we're trying to figure that out. So right now it's more like a preventive strategy instead of like an invention or intervention. Yeah.
Okay. Well, these were two fantastic talks. Thanks, Alex and Sui. And we have one more lecture this year, December 1st. Tom DeLong and Michael Stitzel will speak December 1st at 2 o'clock. And then we had one postponed session. Jonathan Flack and Ebony Carter are going to speak on January 5th. The Pathways program team is going to meet now. Just you all can leave, but we're going to meet. And we may have some other announcements of other future activities for Pathways shortly. But thank you again for the speakers, and everybody have a great afternoon. Thanks a lot.
And Chris, I know that Chandra sent another Zoom link. So I don't know if Marlon, Chandra, you guys just want to hang out here instead of switching over.
Well, we can cut the recording off.
Yes, I can
then
stop
for
that.
sure, because I think that'll be the big thing.